This is a production of Cornell University.
And I did change the title of the talk a little bit because I don't agree that Dandelion is a notorious weed for us. It's one of our favorite plants. And but anyways, that's not the main point of the message, I guess. So thank you so much. So yeah, I would like to tell you a little bit about what we do in my group, which has an alias called Biological Forms and Function Lab, and this is because we are really trying to understand the relationship between the shape that plants have and why they would have that kind of a shape. Um, so yes, we are interested in how plants make their own shape, the form, morphology, or structure in general, the body, like the entire sort of design features of the body. Um, and that's the plant development, isn't it? The questions of a plant development. But I am, as Adrian said, my background is plant development. But the more you get into it, at least for me, I start to wonder, why does it matter so much for the plants to have that particular shape? And so the why element, which is more about make the function, that some kind of a structural function that probably gets targeted in evolution to be selected toward. And so that why it become quite important for me to address once I become sort of independent, or probably I always wanted to address that, and then here's a chance, you have your own group. So that to address that, the form and the functional relationship, I particularly like this sort of sets of phenomena that allows a very my group really likes morphing structures, the structure that change their shape, their structural properties in response to particular environmental or other input, that's certain trigger, because that would environmental stress causing the changes of a shape, that plants do that in so many different ways, and that gives us a nice grabbing point of, right, there's this stress, so plants change the shape, the function probably has something to do with dealing with the stress. And so one of such structures we study is a plant shoot, and the other structure is dandelion diaspora. So dandelion diaspora is something I hope you are all familiar with. I played around as a kid. That you blow at it and then they fly. We chose it because definitely there's some interesting form and function relationship there, and we probably don't know enough about it. That part of that their form and function relationship actually well didn't look like we understood well. But so dandelion diaspora is one of the probably nature's iconic flyers, probably one of the best passive flyers in nature. So they are passive flyers, meaning they catch the wind and fly. They don't use ATP or muscle power to aid their flight. It's a completely, sort of passive. All dead cells. So this structure called the papas or plume, it's a bundle of hair basically. Um, they are thought to enhance drug and dogmatically believed to act like a parachute. However, if you look at the structure in detail, that's quickly sort of crossed out as a possibility because it's basically empty space. It's 90% empty space. This bundle of hair. So the question was, how does this, highly porous disc-like, disc-like papas they can aid flight of the dandelion seed so well. So when you want to understand the function of a plant structure or any structure, often it comes down to fluid mechanics, because fluid mechanics is the field of study which untangles how a structure interacts with its media. In case of plants, it could be the air or water or even soil if you are studying a root. And so in case of a dandelion, that would be the air, and that's the body would be the papas. So we wanted to study fluid mechanics. But for that structure to fluid interaction, basically. And so the starting point is really to get to know the structure. So we did quite a bit of the 3D scanning to capture the geometry, but also mechanical properties, measurement, looking into biochemistry of those papas and so on and so forth. And here is the micro CT scan that we did of the diaspora and the papas. So the papas consists of about 100 filaments, hairs, however you want to call it, and a plus minus 10. So it's quite sort of a consistent, actually. Length of the filaments varies quite a bit depending on the sample, depending on where they grow. But our samples are consistent enough, and it's about 1.5 centimeter across the diameter of the disc. And they are not completely flat. They are spread around, across the 3D, but sort of confined to the range of 2D-ness. And then so this kind of a characteristic we did capture, and we were wondering what aspect of these design features, structural designs, are actually key to the dandelions such a sort of a successful flight. We did a lot of numerical analysis, and as I said, the structural analysis too. We did quite a bit of a characterization, but at the end, the proof of pudding in fluid dynamics is really you look at the flow movement. And so we built our bespoke dandelion size, dandelion design wind tunnel. So it's vertical because dandelion flies vertically, and then we popped the diaspora in, and this is what we saw. If we are to fix the papas and then do the long exposure, this is the picture we get. So surprisingly, that most of the flow, even though it's 90% empty space, most of the flow goes around the entire papas. And but little bit of the flow goes through, but that creates the pressure difference. The pressure in above or downstream of the papas is lower. So that because of that flow around the papas goes inside, and it actually circulates and then kind of stays on for a while in there. So there is so this is a longitudinal, like median section, if you will. And so you can imagine this is in 3D. It's a halo-like a ring of decirculating air, and that stays at the constant distance. It doesn't go away. That kind of air bubble, some people call, or wind wheel, creates regions of low pressure, and physics dictates any object likes to move toward the areas of down the gradient of pressure. So this could explain that the papas and then the seed attached to it would move toward move against the gravity, I guess, toward the low pressure zone, and this could explain why dandelion diaspora can fly so well. So this happened to be the first time the vortex ring, which is detached from body, was observed ever. It was theoretically proposed in a fluid dynamics textbook, but it was never proven or seen in the lab or in nature. And so that was exciting to my fluid mechanics collaborators. But also, so it happens, it turned out to be a new flight mechanism as well. And the new flight mechanism wasn't discovered in the last 50 years or longer, apparently. So that also caught the attention of, sort of interdisciplinary researchers and naturalists alike, I guess. And so what is going on? Why don't the air go through that plenty of empty space that exists in the papas? Well, so there seems to be something called the wall effect at work here. That when there is a porous structure like papas, and then that's small, and that is actually experiencing a very slow movement of media. You can, that air media is very viscous, and so they don't really go through the pore as efficiently as in a bigger pore or the faster flow of flow. And so what happens is, so that porous structure can almost behave like a solid structure. And that seems to be happening with Dandelion. And so this is a diagram of permeability in x-axis, and this is the drag coefficient in y-axis. And what you're seeing is, so that permeability can increase, but actually drag doesn't change from a solid disk for really long time. And then at some point, it's just too porous. Suddenly, this effect, the wall disappears, and then boom, all the air goes through. And this is sort of that more visual diagram of what's happening with something like a 2D disk, porous 2D disk, so like Dandelion papas. And you can see that the mostly solid one looks like this, and then that vortex is attached, and then if it's too porous, about 95% porosity and so forth, so that vortex doesn't form because all the air goes through that disk. And this is the happy, sort of happy Dandelion state. That it's porous enough, but not too porous. And so the Dandelions, the structure is represented by this line, and the Dandelion's porosity sits right around right before the cliff of losing this wall effect. So we thought, we wanted to identify, yes, Dandelion papas is a very complicated structure, but what aspect of that structure, what design features of that structures are the minimal requirement for the formation of the separated vortex ring? And to do that, we took the recapitulation, rebuilding approach, and we basically tried to make that features that we think are important, just that with completely different material, and see if that's sufficient. So what we used was silicon wafer, and we identified that disk shape is probably important, but also it's all about the porosity. So we made that disk artificial papas with a different degree of porosity, and then looked at it, that flow movement, popped that into our wind tunnel and visualized the flow. And sure enough, that this is the artificial, the fake Dandelion papas with 90% porosity, and that created that flow movement, which is almost very close, not identical, but very close to the natural Dandelion. And it wasn't the case with the other porosity levels. By the way, we couldn't make more than 90%. It was so tricky to make what Dandelion makes so easily in nature. on its own, this was a project. The PI actually went into the clean room at the end because we had to come up with a new ways of making this kind of object, and it broke so easily too. Maybe we made 10, and then 2 survived. Yeah, so yeah, we don't have more than dandelion porosity, unfortunately, but yes. So by having that differential porosity gradient, we could also sort of characterize the phases of the porosity, and also the behaviors of this flow movement, that how that depends on the porosity, I guess. And so that when porosity is lower, this formation of a vortex ring is actually quite easily lost. So steady is a steady formation of the vortex, and then that can lost to unsteady, really shedding kind of vortex behind. And that's not good for flight. If it's like that, what happens is, like, you can imagine a coin dropped, they tremble and then they fall very easily. But if the vortex is very stable, then that does aid this, that for the object, the 2D disk to stay sort of aloft. And so the dandelion samples, the natural samples are represented here in blue, and they all fall into this steady range, steady range object, I guess. And I do want to emphasize this because I'm gonna come back to this diagram. Because, as you notice, that those blue dots are quite close to that border to the unsteady range. Okay, so just when we are starting to understand the flight mechanism of the dandelion, we had a visitor in fluid mechanics whose specialty is stability of vortex, and he really wanted to get into that kind of analysis of dandelion papas and then the flow around it. And so what he did was he changed computationally the design features quite a bit, and then simulated that flow behavior, but also the pressure gradient, and so sort of giving us some ideas of what might be the most preferable, most powerful aid for the flight, I guess, like that if it's stable, but also the pressure gradient is strong, and that would be the most favorable. And what he found out was that porosity is a key, but he also found out that the numbers of filaments, that being about 100 was the key. If you change that too much, then that there is still separated vortex ring, but it's not as robust. The vortex, that its effect is not robust. And that was interesting because when you look into the literature, what we find in the counting of filaments, number of hairs in papas of dandelion grow in UK, France, or Japan or China, it's always quite consistent. It's like about 100 plus minus 10. And this might be because, it is quite, sort of a critical for the flight function. Considering my background is a flower development, I do wonder how dandelion controls, make sure that it has such a sort of a specific number of hairs in the papas structure. And so that papas is actually a modified sepal, and it's about 100 filaments, but probably the molecular mechanism, developmental mechanism of making of sepal is the underlying, it's underlying their developmental mechanism. And so from making of few sepals, which are leaf-like organs, to making of 100 like more like hair-like organs, what happened there in the evolution, and also how do they control the number of these highly modified sepal organs? That is a question, and we might be able to guess based on what we know from model organisms, like how sepal numbers are controlled. But that's something, if we can tweak around to alter the number of papa's hairs and then see how that affects flight, that's definitely something we are very interested in doing. However, doing that kind of developmental modulations, re-engineering in Dandelion could be quite challenging. We haven't given up on it, but this is one of the reasons why we don't only work on Dandelion, we work on Arabidopsis as well, and then address similar kind of questions. But and actually do genetic engineering, re-engineering of the structure and the shapes, and then see how the function changes, and so on and so forth. But the Dandelion. Today I'm gonna just talk about the Dandelion. So the reason why we were so sort of keen to study Dandelion diaspora, papa's function, is not only that because it wasn't completely understood why they fly so well, but also this observation that a lot of people noticed, that Dandelion papas is not a static structure. It responds to the environment, specifically the moisture level in the air. And if you drop like 1 microliter of water at the base of the papa's hairs, within 5 minutes or so, it closes. And this is that kind of a morphing observed in nature. So in sort of a misty morning, which was very, very common in cities like Edinburgh, every morning was like that, and they would look like this. And then by the time, like early afternoon, the air was dry enough, so they open up again, and then they go back and forth and back and forth. It's completely reversible. And so we set up, our own mist chamber, and with mists, it's slower. The morphing is slower. It takes about 60 minutes, but it's again completely reversible as well. And this morphing, environmentally induced morphing, probably have a functional significance, because once that morphing, this is not full morphing, it's a half morphing. It's a longer story, but that water droplet sort of dries very fast in the wind tunnel, so we couldn't keep them like completely morphed. But anyways, even with the half morphing, what you see here is that the vortex ring is much smaller, and also it's very asymmetric, and it's not a stable structure. And then that's something you can see in this diagram. So when I talked about this steady phase to unsteady phase of the porous disk in making a vortex ring, that basically, so that the blue dots that you see in this diagram is the wet sample, and then the red dots are what I showed before in the earlier graph, that they stay within the steady range. So that wet, morphed, closed papas is not a good flyer. They don't make that vortex, steady vortex too well. And it's also it's also the case that closed papas diaspora flies faster, that falls faster. So the fading velocity is higher. So we wanted to also understand how that would affect the diaspora function. a more like real sort of a biological setting, which is that purpose is there for the longer distance dispersal. And how does that affect, how does this morphing affect that function, that long distance dispersal? And we try to do this in the field, capturing the wind directions and the speed and moisture level and everything, and then try to follow the diaspora that gets dispersed. And maybe you can imagine it was really, really difficult. Even measuring chaotic sort of nature of actual real wind was proved to be very challenging. So we resorted to computational simulation sort of approach, and we use a world model, which is widely used to calculate the likely distances of flying seed, its dispersal distance based on the terminal velocities and also the meteorological data available to plug in. So what we used was there are such data available for past 30 years from many different cities around the world. And so we chose about 10 European cities, mostly from UK, to see how the dandelion seed would fly in their sort of normal seasons of growth. And so here we have 4 lines, but open and closed, dry and wet, that's that natural morphing seed. And we plotted that. So when the moisture level is high enough, and that's when we say that's the wet phase, wet sample, and then we calculate as such with that terminal velocity and detachment speed, and so on and so forth, so plug it in. And then so we want you to look at the comparison of a green sample with the orange first. The orange is open all the time. That means it doesn't move no matter the environmental conditions. And so this would allow us to, probe the importance of the environmentally sensitive morphing. And the left panel is the all samples, and then the right panel is the best flight, top 1% flyers. Because long distance dispersal tend to come down to those outlier best flyers. So we wanted to capture those two sort of levels of the populations. And so if you look at green versus orange, it's actually not that different. The best flyers is not that different. And we were a bit scratching our head. Oh, right. So no, it's not really. It doesn't help them to go further. And then sort of we thought about it, and we actually divided that natural, the green sort of data to wet condition samples versus dry condition samples, because it's up to one individual diaspora that when they might assume, when they might take off is dependent on those conditions. So maybe we should focus on this separation. And then when we look at it, it was very, very clear that in the wet conditions, that seeds don't travel that much at all. They stay in their own niche, the original niche where the parents, were. And the dry samples actually do go far, and actually they do go farther than the whole sort of population level. The best flyers. And then we thought about it, and then actually realized that it's not like it's better, always better for the plants to go as far as possible. Long distance dispersal is very risky. Maybe they don't find a better niche than what their parents were successful enough to reproduce. And so maybe if the conditions are favorable enough, you do not want to do long distance dispersal. And we start to look into such a possibilities in the literature, not just dandelion, but other species as well, because when we discuss this kind of a possibility, that we found quite a bit of literature in animals. This kind of like environmentally or biologically informed sort of a dispersal based to increase the chance of running into a better or like favor habitat is called informed dispersal. That's quite well known or established concept in animal sort of studies. But for plants, at least quite a few people told us that no, it's that in plants it's really sort of thought to be passive. Dispersal is thought to be very passive event, and so but as a being a plant developmental biologist, that didn't really sort of sit well in me, because I know how sort of resourcefully plants sense the environment and then change themselves to increase the chance of survival and a better, reproduction. And so why would they just be completely passive in such a critical phase of their life? And then so yeah, that's why we looked into other examples of a informed dispersal, if you will. And sure enough, there are quite a bit of examples actually in plant kingdom as well. Like some of the species even make two different kind of diasporas depending on the environmental condition. If it's a harsh condition, dry condition, they make the wing and then they fly further. But if it's a nice wet condition, for example, it just makes like nice mucilage to just, ready and go, run and then start sort of germinating as soon as possible. Or this Asteraceae species, it doesn't even make a papas unless it's a stressful dry condition. And so we think that informed dispersal does happen in plants, and also that the dandelion might be one of such example. And yes. So, and in the remaining time, I would like to tell you very quickly, yes. So what happens, what makes this morphing possible? So the morphing came down to, so we looked at, different parts of this papa structure, what is actually mediating this morphing, and it came down to this base structure, the floral podium. Floral base structure, which is, by the way, completely dead by the time of this, diaspora dispersal. And then so here you see SEM, and this is environmental SEM. That it's getting wet and wet and wet. I hope you can see the hair moves. And yeah, those are plucked off hair bits. And it's quite hard to see what's happening, but if you can see the sideways, this is a cut, in the middle. This sample is cut in the middle, so you can kind of see what might be happening a little bit more. But basically, oops, yeah. So in the dry state, oh, by the way, so this is again cut in half, and then there is a cavity that where the piece used to be, they're dried out, and then it's a empty cavity there. So it's a ring-like structure, but it has the cap of former nectary kind of a tissue on top. And there's a vasculature right in, at the edge of this cavity. And then there are like former cortex tissues that's on the side. And basically this cortex tissue expands sideways. So this crease disappears and then deforms between these cycles, and that changes the angle of these 100 hairs attached to it. And so this is a completely dead tissue, just the cell walls remaining, but different parts of it is expanding and shrinking at different rate. And so this is that quantification of that the vasculature doesn't shrink or expand much at all. And then the cavity actually also doesn't change the shape so much. And yet these side regions, that cortex regions, they expand quite a bit. And this is sort of a diagram. Actually, except for the vasculature, all other cap regions or the side regions, like they expand more or less the same way. And then so more characterization of the movement of the expansions that we did, anisotropy and so forth. And we also, looked at the material composition as well, because those are all polysaccharide plant cell wall, and how can they have differential, expansion properties, swelling properties, because this expansion is induced by the absorption of the water. So we looked at the material composition as well, the chemical composition as well, and then noticed that the base of the papa's hairs have some special sort of lipid enriched sort of a phenolic compound. Phenolic compound was everywhere, by the way. It's very autofluorescent as well, so the entire structure is very much sort of a it's a phenolic enriched structure with a thick secondary cell wall and so on and so forth. But yeah, there are definitely distinct domains at the side and also top, where the former nectary used to be. There seems to be a specialized phenolic compound there as well, which changes the intensity at a different pH. And so we think that is likely to be ferulic acid, for example. And so we, sort of identified there are about 4 different material domains of this structure that seems to swell when they absorb water differentially. And so again, we wanted to know what is the minimal requirement for this structure actuator of this morphing to function as such. And so to do that, we collaborated with Aristide Baudou's group in France, Lyon and Paris. And what we made was a simple 2D axisymmetric FEM finite element method model with these 4 different domains. And we actually plugged in the measured properties into the model, and then looked for the reference model that recapitulate the actual, sort of expansion tendencies and the movement of the papas that best. And that was the trickiest process, but yeah, we took a lot of precautions to do so, and then identified one reference model. And then once, I think the time is tight, so I'm gonna just speed up. We did do a 1 at a time parameter sort of space exploration to identify that this reference model, is a good reference model, but also what particular parameter is important. For example, geometrical parameters, this that cavity, how fixed they are is quite important. If we make the bottom bits sort of free, basically the bottom bits kind of move on top of each other, and that changes the angle of morphing very, very much. So yeah, for example, that actually ended up being the boundary condition we needed to fix that to have a proper reference model. But it really seems to be coming down to a different swelling parameters that these different 4 different domains have that matter a lot for the morphing. And with a digital, sort of a structure that we can do digital mutagenesis. It's very easy to let's fill the cavity and then see how important the cavity is, and then fill it, and then sure enough, without the cavity, that morphing is very much decreased. And how about maybe it's all about the top bits of the cortex? You don't need the bottom bits, and so get rid of it. And no, that wouldn't work. That wouldn't work. You need that the cortex being all around. And how about but does it have to be very specific amount of the cortex? Not really. Actually, this is a natural range. If you increase the bottom bits, the cortex cells more, the that doesn't really change the morphing capability. And so, yeah, we can do a lot of those sort of digital mutagenesis. And what we ended up doing is all those 4 domains are different in the rigidity and the swellability. We swapped all of them to all directions, which really allowed us to identify the minimal criteria for this, sort of a structure to work well, which is basically the vasculature. So there has to be this cavity, that's important. And then there is a vasculature that doesn't do much. It just stay put. It doesn't swell. It doesn't shrink. Yeah, it doesn't move. And then the rest of the structure basically swell, and in response to water absorption, and that's it. So it turned out it is a variation of a very commonly found engineering mechanism that plants tend to use. It's called a bilayer structure. That one side expands more, the other side doesn't expand as much, or it could be the other way around, the shrinkage, differential shrinkage, that actuates the movement. But what is unusual about this structure is it's a ring, and a ring-like tube, and that allows the coordination of many, appendages at the same time, which we haven't found an example like this so much, even though we are looking for it. And so if you know some, we are all ears, and we would love to hear. Okay. So I think I am using up all the time, probably. Just sort of, I'm gonna wrap up now. So we are interested in plant forms and a function, but there are so many interesting functions, shapes, and so we are trying to focus on this particular theme that we identify across the biological world, and also different scales, that we are interested in hair-like slender body structures that's actually really common in plant kingdom, of course, but also beyond. And plants basically are, for me, a composite of cylinder, like sort of slender bodies, stems, and roots, but also hairs on the surface of those larger structures as well. And so it's multi-scaled. And what is interesting about the slender bodies, like hair-like structure, is that they can confer many different structural functions. Like that's a very good way, like making a hair is a very good way to enhance the amount of the ratio, the increased ratio of the surface area to volume. And so it's good for exchangeable moisture or chemical compounds between the outside and the in, and good for mechanosensing, bending elastically, and so on. It could be prickly and good for defense. And when they come in many copies and have, more sort of a haired surface, then that functionality just explode with. And what's interesting about them is also that they tend to have a multiple function. Like in case of a Dandelion papas, which is a very haired, functional structure, it's good for packing. They can pack. They have a lot of empty space among themselves, so they can pack. And it's good for flow control, but also it's good for water capture, which I'm not going to talk so much about it. So far, we haven't been able to really convince ourselves that's biologically important, but they are very good at catching water. Okay, I think I'm going to sort of stop here, but yeah, that hair-like structures you can find everywhere across plant kingdom. And some functions are very scale specific, and some function probably are across some scales, and then that's also interesting question point as well. So yeah, we are interested in why question, the structure function question, but also how, that once we know that's the important sort of structural features, then we want to focus our attention to the developmental mechanism of that making of that structure. So I think that's it. And this is the summary of what we do. But I talked about it, so I'm going to skip to the most important slide, that who actually really did the work that I talked about. A lot of thanks goes to the past and present members of the group. I really show the picture of the main people who did the work I talked today just now. But like we collaborate a lot with the experts of different areas of our inquiry, because we are plant developmental biologists, and but what we want to do, fluid dynamics fabrications, that dispersal modeling, fluid dynamic, sort of the wind tunnel assays and so on and so forth. So for the work I talked about today, we collaborated very, fruitfully with Ignacio Maria Viola's fluid dynamics lab, and they are sort of these members of their lab a lot. Aritsky Buro's group at Lyon and Paris, the members of the group, and a Glasgow, wind tunnel team, and also the dispersal modelers. I showed today, actually we tried ourselves, but we realized no, we need the expert advice. So we reached out to Meryl and James, and so we are doing a better, more realistic modeling at the moment, and then that's that. So before I shut up, I would like to really show who drew that project that I talked about today. So Kahar Cummins, he is an applied mathematician slash fluid mechanics researcher. He made the wind tunnel and then did all the flight mechanism studies. That he led that effort. It was a team effort very much, but he was, the driver. And this is Maddie Seal. She's a very talented quantitative plant developmental biologist who really sort of drove the morphing project. And with that, I would like to stop, but I'm going to leave this one on. So my group has been trying very hard to establish a synthetic biology kit, tools, protocols, and platforms, because we want to be able to do biofabrication to change the functional structures, and that's usually pathway engineering. It's not a one single gene engineering, and that's how we went into synthetic biology areas. And we've been making a lot of tools, and we hope that what we are making are useful to many others around the world. So I'll leave this here in case you are intrigued by one or two of them. Right, with that, thank you very much for your attention.
Thank you, Naomi. Really fascinating talk. There's already a bunch of questions in the chat, so I can read them out to you.
Can we ask our question in person?
Sure, go ahead. Okay, if people have their questions in person, you can also raise your hand. So go ahead and ask your question.
Okay, may I ask my question right now?
Yes.
Okay, thank you. Thank you for very cool and interesting presentation. I like it very much. But I have a question. Okay, what was the relationship between the density of the papas and the velocity? It means that the higher papas, it means that the higher velocity, or no, lower papas has a higher velocity? Do they have a, what was the link between them?
Yes, so the density of the hair, you mean probably.
Yes.
Papas is open, that's sort of lower density, more porous, and that's slower descent. And when it's closed and then the papas is less porous, it falls much quicker, which could be due to, well, they did catch some water. So we did measure that. It's a very minuscule difference, but it might, it cannot explain these changes in the that changes in the velocity of falling completely, but it would contribute. But the fact that it has a smaller parachute, and when the porosity is lower, the vortex that is likely to be helping this flight is not forming. And so there are lots of different aspects to why the closed form doesn't fly as well as opened, more porous form. But we think, that this probably has a lot to do with it.
Okay, I mean, when they are open and has a higher amount of hair, higher number of hair, in one hand they are heavier, okay? On the other hand, they can break the air resistance and move better. They can break the air friction. So my question is that the higher number of hair increase the velocity or decrease the velocity?
So it's not a higher amount of a higher number of hair because the hair number stays the same, but it's the porosity that changes. So when they are closed, that they are more closer to each other, so it doesn't have as much porosity. And you might think that would help that, sort of acting as a wall better, but actually it seems like it doesn't really matter as much for the wall effect, because up until quite high permeability, the wall effect seems to work as well. It seems like, at least from the numerical analysis. And the fact that it's closed so much means that parachute, that the area of the disk is smaller, so it's not really better for flight.
Oh, I got it. Okay, got it. Okay, thank you so much.
No problem. Thanks.
Hello there.
Hi.
Very nice talk, Naomi. I really appreciate it.
Thank you.
I have a question that came up after teaching years of plant taxonomy and looking at dandelions under the stereoscope. And I have a picture there behind me.
Yes.
From one of my dissections. I got to get out of the camera here. But you can see that this region, oh, my finger's too big. Oh, here we go.
Yeah.
This is the mature Cypsella, and this is the flower. But look at this region right in here. This elongates hundreds of times.
Yes.
When it develops. And I was hoping you were going to talk about that today, because biomechanically, that must be a very interesting phenomenon. It goes from a few cells in that region to what happens? Cell expansion, and then some kind of a drying mechanism where it becomes a very rigid pole.
Yeah.
Right?
Yeah, absolutely, Dan. Thanks for bringing it up. That entire sort of development of these structures, especially kind of a past development that completely, changes the shape, like very rapidly, the elongate the stalk, both stalk and also the papa's hair proper. They suddenly elongate, and then it's hollow inside, very, very thick secondary cell wall, highly lignified. And it happens kind of a sort of a when the structure is drying in a, no, that's not true, sorry. Yeah, it's not about developmental patterning, it's a purely control of cell elongation. And yeah, so it's fascinating. And this talk, we didn't talk about it. I didn't talk about it, and we haven't published anything about it, but it is definitely, it helps to center that sort of orientation of the seed. So dandelion seed is rarely like sideways flying. It's always pointing, down. And what's interesting is if you, sort of drop the seed upside down, like immediately it reorients to, the right, like it's almost instantaneously.
It's like a cat.
Yeah, exactly. Yes, yeah. So that stalk is definitely important for that. Also, while flying, it really puts the seed, straight down. However, we also looked at other Asteraceae species which doesn't have the stalk, which are many actually.
That was my next question, because the genus Erigeron has the pappus but no stalk.
Exactly. And when we tried this upside down drop, that they were as effective.
Oh,
Yes. So we think it's more like during the flight, it's actually very good at keeping it, sort of
mhm.
vertical. But yeah, that's definitely one of the remaining interesting question. Yeah. Yeah.
Thank you. Biomechanics of plants, isn't it fantastic?
What can I say? Can't agree.
Naomi, I looked up the identity of the sepals, right? These papas are supposedly derived from sepals. And there's a paper just last year by Vij Verberg, 2021, in Plants, that they use some evo-devo approaches. And it is homologous, but it's a very weird, highly derived version of it that forms these. So there's some interesting developmental issues regarding the floral whorls and the identity of the genes that are involved in those structures.
Yes, and it's a spirally or sort of a position. It's about 3 rounds of a spiral. So
They
it's
said
not
it's from the inner floral whorl. Inner.
inner. Okay.
That's what they said. Which sepals should be the outer floral whorl. So I need to look at that paper a little more carefully, but
Yes.
that's kind of wild.
Yeah, that is indeed wild. Yeah, but this is a weird, wild structure, I must say. But it's
Yeah.
not common though.
So maybe our developmental people can inform me about what that paper is telling us. Jeff's got his hand up there.
Yeah, I would go back. Hi, hello.
Hi, Naomi. That was a very nice talk. I have a question. I'm interested in polyploidy. And of course, Taraxacum officinale is a polyploid series with triploids and diploids. And I'm just wondering if you've looked at natural variation for these structural characters of the pappus and whether or not cell size is affected. And if so, how does that affect downstream phenotypes that might be relevant for dispersal and for the biomechanics you're studying?
Yeah, thank you. So as I mentioned, the number of papa's hairs seems to be quite fixed, no matter, different country, even like completely different system that people looked at. However, the diameter or radius of this papas varies a lot, even on the same plant. I think it actually varies. And that is a bit of a question mark how much it affects, because actually that sort of a functional domain of the papas is not entirety. That beyond certain sort of midpoint or so, the distance between 2 hairs become too wide, so they actually goes beyond the cliffhanger of the wall effect. So it's actually the inner bits that's doing the most of the job. So actually these changes might not matter so much for the flight function, that diameter change. However, it's very possible they don't have this morphing when they grow in a different ecosystem and so on. We are collecting samples from different sort of environment that dandelion has been growing, and also we also made the dry treatment samples as well for 3 generations or so, and then we are sort of looking at it too, and definitely those are sort of interesting. Yeah, some of the really interesting questions for future, but surprisingly, it's so variable in a given habitat as well, and then very plastic, I would say. So yeah, how much of a fixed, very clear sort of a ecological sort of ecotype sort of a difference that we can find, etc. I think it comes down to, can we find a really good samples that really is stable, and then that sort of embedded into sort of that where the habitat, and then we know where they come from, and so on and so forth. So I'm not sure if I answered.
Well, I'm just curious, do you know the ploidy of the individuals that you've been using for your modeling?
Yeah, so we collected these samples in a meadow, like 10 meters away from our lab. We can go back there and then we collect it, but actually even that, so dandelion have about 500 known subspecies. And then trying to identify the subspecies, to be honest, we haven't succeeded yet. It's so subtle that we are consulting one of the few, UK dandelion expert. But even they say, no, we have to see the field grown leaves from May to June or something like that. And then so we haven't been able to identify, but we have the samples. We have the sample we actually propagated in the greenhouse for 3 generations, and then have a very uniform, phenotype, etc. So if we want to characterize their ploidity level and stuff, we do have a very good collection of that sample. So we can do, but we don't know yet.
Okay, thank you very much.
Thank you. Should I answer the questions in the chat? How quickly do the seeds germinate after the drop to the ground? Is it very quick? I was wondering if they are dropping because the conditions are optimal for germination. Yeah, so dandelion seeds in our hands, it was very difficult to germinate them. Putting on the wet soil simply didn't germinate them so well. We actually had to keep them in water immersed for a couple of weeks. So our feeling is they really like wet condition for germinating. That's as much as we know. But that does go hand in hand with, the preference for wet environment to fly into and things like that. How much natural variation is there in dandelion papa structure? Yeah, so I ask because taxonomic scenario occurs as diploid sexual individual. Yeah, and triploid apomix. So this one is apomictic. Yeah, it is. Yeah. So maybe that would answer your question, maybe, Jeff. But yes, that we know. We did confirm that since cell size often correlates with ploidity. Yeah. Cell size, but we do look at the whole structure, and then that's what matters. But thank you for that. We really should be calling these fruits instead of seeds. Yeah, it is seeds. Yeah. So sorry, fruits. Absolutely, it's ahin.
That's my comment. I'm being a technical botanist on you.
Ahin, and then that papas bits is actually appendage. It's not even part of fruit. So we did go through, how should we call it? And diaspora, I don't know why. A lot of people said
that's too general. That refers to anything that disperses. So let's be precise. Let's call it a keen. Some people say a keen, but it's derived from a bicarpellate ovary. So people in the Asteraceae invented their own term, Cypsella. So just say Cypsella. That's the word we've used for 100 years.
Okay, great. Thank you. Yeah, Cypsella.
Isn't it a pretty one? Look at that pretty Cypsella.
I like that. It sounds very nice. Yes, Cypsella. Okay, thank you so much. But you resolved our years of discussion. Thank you. I was wondering if air moisture changes, how easily a diaspora detaches from the mother plant. Absolutely. I did not mention this, but seriously, oh yeah, I have this still on. Let me show you this. Do you see? So this is a lateral flow, big wind tunnel, and we put the wet and the dry inflorescence. Sorry, yeah, and then so it's like looked at how easily that Cypsella flies away, and with a wind tunnel straight in the air, like without the turbulence introduced, that it was really hard to detach, and so it stays on the parent for longer. And at some point, though, they seem to just drop. And then that how they do that in the nature, I think we really need to do a bit of the field studies. But yeah, absolutely, I completely agree, and that would be a very interesting study on its own as well. Such an interesting parallel with Casprea fruits. Yeah, deficiency that occurs through differential dissection of different tissues. Yeah, located in different places in the fruit. Yeah. Are you thinking that it's something like a Kadamine and things like that? Yeah. Thank you. Yeah, there's a lot of strategies and clever engineering mechanism in how plants disperse and yeah, absolutely. At least I am fascinated by that. Thank you. Thank you so much for the questions. I hope I answered most of them.
This has been a production of Cornell University, on the web at cornell.edu.